Hello, my dear students. It's been quite a time. I want to welcome you to the class of BCH 407, Biochemical Pharmacology, once again. We are in lecture 9 in the series, and the topic is mechanism of drug actions. I take it again. The topic is mechanism of drug actions. What do we expect to achieve at the end of this lecture? We have 5 points in mind. These are that at the end of this lecture, you should be able to define the term mechanism of drug actions. Secondly, describe 4 main types of drug targets, such as receptors, enzymes, ion channels, and transporters. You should be able to explain the concepts of agonists, antagonists, partial agonists, and reverse agonists, as well as interpret those response curve and discuss potency and efficacy. Now, starting by way of definition, what is mechanism of drug action? It simply refers to the dynamics of ingested bioactive substances with specific biochemical targets such as receptors, enzymes, ion channels, transporters, and non-receptor molecules to produce the desired therapeutic outcome. This concept of mechanism of drug action is central, and it is crucial for understanding how drugs achieve their intended therapeutic outcomes as well as their potential side effects. Now, you want to know what are the 5 mechanisms, or how do drugs achieve their desired effects. That question can be answered by considering the 5 major means by which drug interacts with substances to achieve the desired effects. One of such is receptor interaction, enzyme modulation, ion channel modulation, transporter interaction, and non-receptor mechanism. We are going to look at each one of these in turns, but before I go into it proper, let us look at the surface of these 5 mechanisms. Receptors can be of various types, including inotropic, G-protein coupled, enzyme linked, or nuclear receptors. Enzyme modulation involves the activation of enzymes, thereby altering the rate of biochemical reactions. Ion channel modulation involved opening and closing of ion channels in membranes, thereby affecting movement of ions in and out of the cell, thereby altering cellular activity. In transporter interaction, Some drugs affect transporter proteins, influencing the movement of substances across cell membranes. For non-receptor mechanisms, it should be noted that not all drugs act through specific receptor or through receptor at all. Some, like antacids, work by neutralizing stomach acid through simple chemical reactions, which you shall see later in the lesson. Now let's start looking at these items I mentioned earlier, starting with drug action through receptor interaction. Please note that nuclear receptors are the same as intracellular receptors. Of course, there are two types of receptors, intracellular and surface receptors. The intracellular or nuclear receptor is found in the nucleus, while the surface receptors are found in the cell membrane. Now, the intracellular or nuclear receptor functions to bind lipophilic drugs, such as hormones that regulate gene transcription. Now, let's go ahead and describe the nuclear or intracellular receptor. From the diagram before you, I have two figures here labeled A and B, the active, inactive receptor as well as the active receptor. In its inactive state, nuclear receptors consist of the following components. You have the N-terminal, the beginning of the receptor protein, transportation activation domain, responsible for receptor transport and activation functions, DNA binding domain. This domain enables the receptor to bind to specific DNA sequence once it is activated. We have the ligand binding domain, a site within the molecule that binds a specific ligand, such as hormone. We have inhibition protein, which bounds the receptor. This protein keeps the receptor in an inactive state by blocking the ligand binding domain or site. We also have the C-terminal, which mark the end of the receptor protein. Upon binding of a ligand to the receptor, it causes conformational change to the receptor, leading to release of inhibition protein. The receptor can now bind to the specific DNA sequence known as the hormone response elements, HRE. A co-activation protein binds the activated receptor, facilitating transcription activation. The receptor co-activator complex interacts with the target gene, initiating gene expression. This gene expression leads to the production of a target protein, which executes the physiological response to the hormone. Number 2, action of receptor or action of drug through receptor interaction. Now we are looking at G protein coupled reactors or G protein coupled receptors. It functions to trigger second messenger cascade inside the cell, as depicted by the diagram before you. This process begins when an external signaling molecule or a ligand, such as a hormone or neurotransmitter, binds to a GPC arrow embedded in the cell membrane. This receptor spans the membrane and transmits the signal through outside of the cell to the inside. Upon ligand binding, the GPC arrow undergoes a conformational change that activates the associated heterotrimetric G protein composed of 3 subunits: alpha, beta, and gamma subunits. In its resting state, the G alpha subunit of the protein is bound to GDP and remains associated with the G gamma beta dimer. This conformational change in the GPC arrow promotes the exchange of GDP for GTP on the G alpha subunit, thereby activating it. As a result of these happenings, the G alpha GTP separates from the G beta gamma complex, and both components can independently react with different intracellular effector proteins. In this active GTP bound state, the G gamma subunit and the G beta gamma initiate distinct signaling pathways by modulating enzymes and ions, thereby producing a range of cellular response. The signal is terminated when the G alpha subunit possesses intrinsic GTPase activity, which is often accelerated by a regulator of G protein signaling proteins. The GTP bound to G alpha is hydrolyzed to GDP, converting G alpha back to its initial form. Following this, the G alpha subunit reassociates with the G beta gamma dimer, reconstituting the inactive protein complex. Now let's take a look at the tyrosine kinase receptors. This is a little complex but straightforward. We have here a sequence of activation and deactivation processes. The first is receptor tyrosine kinase. Before signal binding, you can see the binding domain is empty. Now, upon the receipt of signal, that is, binding of a molecule to activate the protein, in the activated kinase domain. Signal binding causes dimerization of receptor and activation of tyrosine kinase domain. This leads to addition of phosphates onto each other in the phosphate binding site. This is followed by complexation of signaling proteins, which assembles on phosphorylated ROTK, that is, receptor tyrosine kinase tail. This complex can activate ROAS. Now, in step 5, here, you see the activation of ROAS. It involves the G protein molecule. And step 6, still on the cell membrane, the MAP kinase is mobilized, activated, and ready for actions. Next is the inotropic receptors. Another term for it is ligand-gated channels. These are the type of channels you find on the neurons. These receptors can only be opened by specific ligands. Neurotransmitters and receptors fit together like lock and key. Only neurotransmitters are able to bind and open certain receptors in the neuron. On enzyme modulation, the word enzyme modulation is used to mean the process by which the activities of enzymes are altered, either increasing, that is by activation, or by inhibition, decreasing the activity by specific molecules, including many drug substances. Since enzymes catalyze biochemical reactions, modulating their activity can change the rate, extent, and pathway of metabolic and physiological processes. Now, how do drug modulate enzymes? It does so by slowing down or blocking enzyme activity, which is called enzyme inhibition, by competitively inhibiting enzyme action, or by non-competitively inhibiting. That is when a drug binds to an allosteric site, causing conformational change that reduces enzyme activity. We also have irreversible inhibition of enzyme. This is when a drug forms a covalent bond with the enzyme, permanently inactivating it. Remember, in the initial stage of this lecture, I mentioned to you that covalent bonds are not really needed in normal cell function, but they are desired in toxicology. In drug modulation of enzymes, enzyme activation is less common, but include drug or molecules that increases enzyme activities, often by binding allosterically or stabilizing the active form of enzyme. An example is AMP activation of phosphofructokinase in glycolysis. The next is ion channel. The diagram projected illustrates the function and gating mechanism of a ligand-gated ion channel in response to the binding of a specific ligand neurotransmitter. Now, from the diagram shown previously, you can take time to go back and look at it. From the resting state, the ion channel is shown embedded in the membrane. The ion channel is closed and no ion can pass as a result of the closure. Ligand binding and conformational change occurs when the ligand binds to the ion channel. It triggers a conformational change in the protein structure. This is indicated by the arrow labeled conformational change, pointing from the top to the left bottom. Upon binding, the channel is now open. The open channel allows ion to flow through. Now the ligand unbinds and resets. When the ligand unbinds and the channel returns to its original closed conformation, ready for another cycle. Now, how does drug modulate ion channels? First of all, let's take time to define ion channel modulation. This refers to the alteration in the behavior of this channel, either by enhancing, that is, opening or blocking ion flow, thereby altering cellular activity. We must know that channels are proteins within the membranes that allow specific ions, such as sodium ion, potassium ion, calcium ion, and chloride ion, to flow in and out of the cell. This ion movement regulates electrical signaling, muscle contraction, secretion, and other processes. Now, what could be the ion channel modulation mechanism? They are threefold. One, they block ion channels, that is, ion channel blocker or channel blockers. Another term for it is channel inhibitors. The second is ion channel activators, that is, another word for it is channel openers. And of course, we have allosteric modulators. The ion channel blockers block ion channel pore, preventing ion from passing. The effect is that it inhibits cell excitability or downstream signaling. For channel openers or activators, channel opening enhances ion flow, and this can increase excitability or activate physiological responses. While allosteric modulators bind to other sites other than ion conducting pores and alter the ion channel responsiveness. Positive modulators enhance channel activity, while negative modulators reduce channel activity. How about transporter interaction? Transporters are specialized membrane proteins that facilitate or regulate the movement of substances, including drugs, across cell membranes. There are different types of transport proteins. We have the channel proteins and carrier proteins that move molecules either passively or actively. Active transporters that uses ATP hydrolysis to move substances against concentration gradients. We also have passive transporters that facilitate movement along gradients. How do drugs interact with transporters? Drug interacts with transporters in several ways, such as being a substrate for transporters where they cannot enter or exit cells, thereby affecting their bioavailability and therapeutic effects. Secondly, some drugs inhibit or induce transporter activity, altering the transport of other drugs or endogenous substances. This can lead to drug-drug interaction by changing the drug concentration in tissues or plasma, impacting efficacy or toxicity. Thirdly, transporter function can also be modulated indirectly by drugs through regulatory pathways affecting transporter expression or localization, which further influences drug pharmacokinetics. Non-receptor drug mechanism. What does it mean? It refers to drug action that do not involve binding to specific receptor on cell membrane. Instead, their therapeutic effect arises from altering the chemical environment rather than interacting with cellular receptors. Antacids are a classical example of such drugs. They work by neutralizing stomach acid by simple chemical reactions rather than receptor binding. Now, example is magnesium hydroxide reacting with stomach acid. It is a basic neutralization reaction which reduces acidity and helps relieve symptoms such as heartburns and indigestion. With that being said, let's look at some common concepts. Activation or inhibition of target receptors are governed by relevant concepts such as agonist, antagonist, partial agonists, and reverse agonists. Again, let's take these terms in turns and have them defined. First is agonist. An agonist is a chemical agent that has the potential to attach to a binding site on a receptor and leads to a response through other molecules such as transducers. Otherwise, the simple binding of an agonist to a receptor will not produce any response. Now, we shall come to the issue of transducers later in the course. On the other hand, an agonist is an agent that can bind the same binding site on a receptor as an agonist, but does not lead to a response. By binding the same site as the agonist, it prevents the agonist from occupying its site, and so no action can occur. There may be competitive and non-competitive or irreversible agonists. Agonists are useful to avoid excessive receptor activation that can cause toxicity or receptor desensitization. Their efficacy can be higher, lower, or equal in affinity compared to full agonists. How about reverse agonists? They bind to the same receptor as agonists, but induce the opposite pharmacological effects. This requires the receptor to have constitutive activity in the absence of a ligand. While agonists increase receptor activity above the basal level, inverse agonist reduces receptor activity below basal, producing a negative intrinsic activity less than zero efficacy. Neutral agonists have zero efficacy and block both agonist and reverse agonist effects. Let's look at the dose response curve. A dose response curve is a fundamental concept in pharmacology and toxicology. First of all, let's examine the axis. The x-axis usually is the amount of drug or compound administered. The y-axis is the biological or physiological response observed as a percentage of the maximal possible response. Now, the shape of the curve. The curve is sigmoidal, meaning S-shaped, typical of many drug dose response relationship. At low doses, there is little or no response. As the dose increases, the response increases steeply in a middle range. At higher doses, the response plateaus, approaching the maximum, which is in fact near 100%. If you do that, the EC50 should be the point at which your curve coincides with the 50% at the y-axis. That would be the effective concentration of the drug that can produce 50% of its maximum effect. What does the EC50 convey to us as scientists? I've told you that EC50 is the concentration at which drug elicits 50% of its maximum effect. In the real sense, EC50 is a measure of drug potency. A lower EC50 means a drug has higher potency. A high EC50 means low potency. You will need to take the same procedure with one or two modifications to achieve LD50. LD50 is different from ED50. LD50 is the dose of a substance that causes death in 50% of a test population. Don't use humans, so they're animals. 50% of them, you get your if 50 of them die, you know the concentration you gave that caused the 50% of them to die. So that's 50, that's concentration is the ED50. IC50, also similar to EC50. Almost the same procedure to determine your IC50 for a particular drug. Let's look at potency and efficacy. Potency refers to the amount of drug needed to produce a given effect, typically 50% of the maximum effect. While efficacy, on the other hand, refers to the maximum effect of a drug can produce regardless of the dose. Now, in this lecture, what have we done or what have we achieved? Now, we have known that drug can act through 5 primary mechanisms, such as receptor interaction, enzyme modulation, ion channel modulation, transporter interaction, as well as non-receptor mechanism. We have learned the receptor types and of course examples such as nuclear receptors, G-protein coupled receptors, inotropic receptors, and tyrosine kinase receptors. We've also known about inhibition, that is in enzyme modulation, and the types we mentioned are competitive, that is bind active sites, non-competitive, bind allosteric site, and irreversible, forms covalent bond. We also know that enzyme activation involves the enhancement of enzyme activity. In the course of this lecture, we learned about blockers and openers. Blockers inhibit ion flow, while openers enhance ion flow. Of course, allosteric modulators, as concerns ion channel modulations, alter channel activity indirectly. We also acquainted ourselves with transporter interaction. We said drug can be a substrate, inhibitors or inducers, and can indirectly modulate via gene regulation. These interactions affect drug bioavailability, efficacy, and toxicity. We have also learned that there is something as non-receptor mechanism, where drugs act by chemical interaction, such as in the case of antacid, neutralizing hydrochloride or hydrogen chloride. We also in the course of this lecture, learn some basic concepts like agonist, which binds and activates receptors, antagonists, bind receptors but block activation, partial agonists, activate receptors with submaximal effects, while reverse agonist induces receptor activity below basal level. Mention was made also of, during the course of this lecture, of dose response curve. It was mentioned that in such curve contains the x-axis, the y-axis. The x is the dose or concentration domain, y is biological response domain. Then we have the typical sigmoidal shape, then the plateau at the maximum effects. We have we mentioned or we learned about the pharmacological matrix such as a EC50, IC50, LD50, potency, and efficacy. Potency, remember, is the amount needed for effects. Efficacy is the maximum effect achievable. Now I have provided below for you, or I've provided here for you, a set of questions that you can use to assess your rating or your standing as far as this topic is concerned. Take time to go through, study, and assess yourself. In case of any difficulty, you reach me through the usual channel. Thank you for listening and good luck.